think I must be on. Okay. Jane, thank you for that really wonderful introduction. I don't think I've ever had such one. And I certainly never before thought of myself as a salmon. But I won't forget that now. Okay, so as Jane mentioned, we're interested in the sense of smell. And through this sense, humans and other animals can perceive a vast array of chemicals as having a distinct odor. All these chemicals are small volatile molecules, but they have different structures, and somehow these different structures are perceived as having different odors. The olfactory system governs the sense of smell, and it is characterized by exquisite sensitivity and discriminatory power. Even a very slight change in the structure of an odorant molecule can change the way it smells. For example, these two molecules have similar structures, and yet one smells like pear and the other one like banana. Now, in mice and other animals, the olfactory system also detects pheromones released from members of the same species and predator odors. And these can elicit innate responses, such as changes in hormone levels or instinctive behaviors, such as mating, aggression, or in the case of predator odors, fear. So how does the olfactory system detect so many different chemicals? And how does the brain translate those chemicals into diverse odor perceptions and innate hormonal changes and instinctive behaviors? Now, mice have two olfactory sense organs, the olfactory epithelium, or OE, that lines the nasal cavity, and the vomeronasal organ, or VNO, in the nasal septum. The OE detects odorants and social cues, and the VNO is thought to be specialized for social cues. Signals from these two sense organs travel through different neural pathways in the brain. OE signals are relayed through the olfactory bulb to the olfactory cortex, and then sent to a number of other brain areas. These include higher cortical areas thought to be involved in odor perception and discrimination, as well as deeper limbic areas of the brain, the amygdala and the hypothalamus, which control hormone levels, basic drives, and instinctive behaviors. In contrast, VNO signals are relayed through the accessory bulb to the vomeronasal amygdala, and then primarily to the hypothalamus, which controls these basic functions and drives. As Jane mentioned, in our initial studies, Richard Axel and I asked how odors are initially detected in the nose. In those studies, we discovered the odorant receptor, or OR family. This family has 1,000 different members in mice and about 350 in human. In later studies, our two labs and others found two different families of receptors in the VNO, the V1Rs and V2Rs. And more recently, we identified two more families of chemosensory receptors in the olfactory system, a family of 14 trace amine associated receptors, or TAARs, in the nose, and a small family of formal peptide receptors in the VNO. Our early studies and others suggested that each olfactory sensory neuron, or OSN, in the nose is likely to express a single OR gene. The nose, the nasal olfactory epithelium, has a series of partially overlapping spatial zones excuse me, that express different sets of OR genes. And neurons with the same OR are scattered throughout one of those zones, but their axons all converge in a few specific glomeruli in the olfactory bulb of the brain. This creates a semi-stereotype map of OR inputs in which each glomerulus and its associated projection neurons, which send information to the olfactory cortex, appears to be dedicated to input from only one OR. We found that the OR family is used in a combinatorial fashion to encode odor identities. Different odorants are recognized and thereby encoded by different combinations of receptors, but each receptor can serve as one component of the codes for many different odorants. Even if each odorant were recognized by only 3 receptors, this combinatorial scheme could generate a vast number of different receptor codes and thereby allow for not only the detection, but also the discrimination of a multitude of different odorants as having different scents. These studies also provided insight into several puzzling aspects of human odor perception. As I mentioned earlier, highly related chemicals can have different smells. Excuse me. We found that highly related odorants with different perceived odors in humans were invariably recognized by different combinations of receptors. So if you change the structure of a chemical, even slightly, you can change its receptor code, thereby leading to a change in perceived odor. We found that changing the concentration of an odorant also could change its receptor code by recruiting additional receptors into the odor response. This may explain why it is that changing the concentration of an odorant can also sometimes change its perceived odor. Now, in later studies, we looked at a larger variety of different odorants, and what we did was to use calcium imaging to look at responses of single OSNs from the mouse nose to 125 different odorants. And individual neurons were exposed to 13 different odorant cocktails containing these odorants, and then retested with single odorants from cocktails that elicited a response. So altogether, we tested 39,000 different combinations of OSNs and odor cocktails in these experiments. These experiments suggested that the receptor code for an odor can vary from a single receptor up to over a dozen receptors. I should mention that we did find evidence that in later studies that indeed most single OSNs express only one OR gene. So although we were analyzing the activities of individual OSNs, those by and large reflect the activity of single odorant receptors. Excuse me. So remarkably, we found that 94% of odorants stimulated a unique set of OSNs in these experiments, emphasizing the amazing discriminatory power of the olfactory system. These studies also indicated that the OR family and the OSN repertoire is characterized by extreme diversity. Individual neurons that responded to the cocktails showed 92 different response profiles comprised of responses as single cocktails or combinations of cocktails. This diversity was also seen when we checked individual odorants. For example, different aldehydes, we saw 36 different response profiles composed of responses to different single aldehydes or combinations of aldehydes. These studies further showed that most OSNs, and by extension most ORs, are narrowly tuned to recognize a subset of odorants with related structures. We did find some OSNs and individual odor receptors that were broadly tuned to recognize a large variety of odorants, and we don't know why, if there is any advantage to having some of those or not. But anyway, most of the OSNs we tested did respond to a specific subset of odorants with related structures. And interestingly, in some cases, such as the two shown here, those odorants have related odor qualities or subqualities. For example, this neuron recognized 4 vanillin-like chemicals, all of which have a sweet odor subquality, and these 3 recognized by this neuron all are macrocyclic musks, and they all have a musky quality. So it's possible that though the OR family is used in a combinatorial fashion to generate different odor perceptions, some ORs may be able to convey a particular odor quality or subquality. We also found a small number of neurons that responded to animal-associated odors. For example, one shown here responded to trimethylthiazolane, a fox predator odor that causes innate fear. Another responded to two fecal odors, indole and scatol, which have a repulsive fecal odor. And another one responded to cadaverine, which has the smell of decaying flesh. So one possibility that intrigues us is that some of the receptors that recognize these animal-associated odors may be able to elicit an instinctive response in mice. Okay, so altogether, these findings indicated that the spatial code for an odor in the nose is a dispersed ensemble of neurons, each expressing one component of the odorants receptor code, whereas at the level of input to the bulb is a specific combination of glomeruli that receive input from those ORs and have a similar arrangement in different individuals. It's not clear how OR inputs are organized at the next level of the olfactory cortex, but recent studies suggest that information may return to being highly distributed at that level. How the organization of how olfactory information is organized in the olfactory cortex and at higher cortical levels to ultimately generate different odor perceptions, however, is still largely a mystery. Now, another puzzle is how the organization seen at lower levels of the olfactory system is generated during development. One question is how the single OSN in the nose comes to express a single OR gene. Now, in a prevailing model of this so-called OR gene choice or OLFR choice, the neuron initially selects a single OLFR allele for expression, and feedback from the encoded OR protein prevents the expression of other OR genes or OLFRs. Excuse me. So to gain further insight into this process, Naresh Hanchade, a postdoc in our lab, who's here at the meeting, decided to conduct single-cell transcriptome analyses on developing mouse OSNs. And he did this in collaboration with a wonderful computational biologist at the University of Washington, Cole Trapnell. Now, it's known from work by Jane Roscombs, actually, and others, that the olfactory epithelium contains different stages of OSN development. The olfactory sensory neurons are replaced throughout life by an underlying layer of progenitor cells. The progenitor cells give rise to neuronal precursors, which in turn give rise to immature OSNs and then mature OSNs. So what Naresh did was to dissociate the OE, the olfactory epithelium, and prepare single-cell cDNA libraries from individual cells, do PCR to analyze the libraries for known markers of the 4 different cell stages, and then use, together with Cole, Illumina sequencing to identify genes expressed in each neuron. Now, using a unbiased algorithm that he had developed, Cole found a linear trajectory of development among the OSNs in the OE. Looking at the transcriptome data for different cell stage markers, it was clear that this trajectory reflected the known progression or the expected progression from neuronal progenitors to precursors through immature OSNs and finally mature OSNs. Now, Naresh has a poster that he's going to show tonight, so I would encourage you to go and talk to him, and you can find out more details about this work if you're interested. Okay, so we found that there are over 3,800 genes that are differentially expressed over OSN development. And interestingly, there are clusters of genes that Cole identified, as showed here, that changed their levels of expression in waves during development, suggested coordinated changes in gene expression that occur during particular windows in development. Now, looking at the gene ontology, using a gene ontology database, Naresh found that each of these clusters contains a variety of different transcription factors, and also most of them contain chromatin modifiers, suggesting potential regulators of OSN development and possibly OLFR choice. Now, OLFR expression among the neurons that were examined, so we're talking about multiple neurons from the 4 different stages of OSN development. Actually, we divided the immature OSN stage into early and late immature OSNs. So looking at the transcriptome data for OLFR transcripts, what we found was that they first appeared at the precursor, or more often at the early immature OSN stage. And the transcript level for OLFRs gradually but unevenly increased over development to the mature OSN stage. Some mature OSNs had 100-fold or more higher level of transcripts than the early immature OSNs. Now, consistent with what was thought from previous experiments, most mature OSNs did express a high level of a single OR gene, although in some cases, some of them expressed lower levels of a few other, 1 or 2 other OLFRs. However, surprisingly, and in sharp contrast to the prevailing model of OR gene choice, we found that early immature neurons can express multiple different OLFRs. And in fact, we identified up to 12 different OLFRs expressed in a single early immature neuron. So Naresh validated these studies in vivo, which can be very important if you're doing single-cell RNA-seq. And in the neonatal OE, which contains many immature neurons, he saw a small percentage of neurons that were co-labeled by a single OLFR probe and a mix of probes from the same zone, OLFR probes from the same zone. And using a single molecule detection technique, he was able to see a small percentage of neurons co-labeled by 2 different OLFR probes. And consistent with the decline in immature neurons during overdevelopment, he saw a higher percentage of these double-labeled neurons in the neonatal OE compared to the adult OE. Now, interestingly, he found that when he looked at the expression of OLFRs that are co-expressed in single neurons in the adult OE, the OLFRs that co-expressed in single neurons were always expressed either in the same or partially overlapping zones in the adult. So this suggested that the developing OSN is restricted to a regionally biased subset of OLFRs. Now, in previous studies, we and others, after the publication of the mouse genome project, we and others found that mouse OR genes, and this is true in humans also, are highly distributed in the genome. We found clusters of OR genes located on the majority of mouse chromosomes, in fact. So one possibility that we entertain with regard to the multiple ORs expressed in single neurons was that chromatin changes at a single OR locus might result in the co-expression, low-level expression of multiple OLFRs clustered at that locus. However, when we looked at the locations of genes expressed in the same neuron, what we found was that OLFRs co-expressed in a single neuron could map to multiple loci on up to 7 mouse chromosomes. So this clearly rules out the possibility that the OLFR co-expression is due to the activation of a single locus. Rather, it looks like the early co-expression of OLFRs is due to an availability of OLFR genes at multiple different loci on multiple different chromosomes for activation by the single neuron. So these studies suggested that the single developing OSN can initially express multiple OLFRs and then progress over development to express only one OLFR in the mature state. To see whether odor-induced neuronal activity might be involved in this transition, Naresh looked at the 4 different transcriptome data for the expression of genes encoding 4 different olfactory transduction molecules downstream of ORs, which are involved in generating the signals produced by odor binding. And these were GNAL, ADCY3, CNGA2, and CNGA4. And what he found was that based on the transcriptome data, OSNs expressing multiple OLFRs can express all 4 of these transduction molecules, and neurons lacking one or more of the transduction molecules can express only one OLFR. So this suggests that odor-induced neuronal activity is not involved in the progression from neurons expressing multiple OLFRs to only one OLFR. We also don't think that the immature OSNs expressing multiple OLFRs simply die, because many of the immature neurons express multiple OLFRs, and studies of apoptosis in the OE by Jane Rouskams and others indicate that there's only a very low level of apoptosis occurring even in the neonatal OE. So we think that changes in OLFR expression over OSN development may emerge via cell-autonomous environment-independent mechanisms. At present, we don't know what those are, however. I'd now like to turn to a different question, and that is why there are TAARs in the nose. The large number of ORs and their combinatorial use in odor coding would seem to easily explain odor discrimination. However, when he was a postdoc at our lab, Steve Lieberlus discovered that the OE expresses 14 trace amine-associated receptors, or TAARs. He found that these TAARs have expression patterns like ORs, as shown here, with different TAARs expressed in different neurons, and those neurons did not express ORs. And it was already known that, like ORs, TAARs are evolutionarily conserved in vertebrates. So these findings together suggested that TAARs may have a function distinct from that of ORs. We found that in a screen for ligands for TAARs, we found ligands for some TAARs. All of those were volatilamines, And interestingly, 3 TAARs recognized amines that were reported to be in mouse urine, raising the possibility that TAARs might recognize social cues, which are abundant in rodent urine. Consistent with this idea, Steve found that one TAAR, mouse TAAR5, selectively responded or preferentially responded to urine from adult male mice, suggesting that this TAAR could discern both gender and sexual status based on urinary cues. Now, subsequent studies by Steve in his own lab at Harvard and by Tom Baza at Northwestern indicated that 2 mouse TAARs are required for attraction or aversion to animal amines. And Seeger and Korsching in Germany found that one zebrafish TAAR ligand causes aversion in the fish. Meanwhile, Lisa Horowitz in our lab found that TAARs are expressed in the macaque, higher primate, suggesting that they also act as olfactory receptors in humans. And interestingly, we and others found that human TAAR5 responds to an odorant, an amine that's very repulsive to humans. And also we found that it responds to rotten salmon, but not fresh salmon, which most people don't like. So together, these studies suggested that the evolutionary conservation of TAARs might be linked to an ability to induce innate responses to amines. To further explore this idea, Luis Saraiva, another postdoc at our lab, first conducted a larger screen to find additional TAAR ligands, and he did that. He found a number of additional amines that acted as TAAR ligands. And he then tested them for the ability to stimulate either attraction or aversion in naive mice, as you can see here. So what he found was that indeed many of the TAAR ligands he tested could cause attraction or aversion, but not all. And curiously, though, he found that ligands for the same TAAR could have different behavioral effects. For example, looking at TAAR5, which was already known to be required for attraction to one ligand, TMA, you can see here that some of the TAAR5 ligands were attractive and other ones were completely neutral. So in thinking about this, we knew that some TAAR ligands activated more than one TAAR, and it was quite possible that many or all of the TAAR ligands can also activate ORs, which were unknown to us. So one possible explanation for this, we thought, was that these results could occur because the combination of because of a combinatorial effect on behaviors. So, for example, like odor perceptions, it could be that the actual behavior was influenced by the combination of receptors activated. So in this model, one OR might be able to, or TAAR, might be able to modulate the behavioral effect of input from another receptor. And consistent with this idea, Luis found that when he presented animals with binary mixes of odorants, some attractive and aversive odorants could modulate or block each other's behavioral effects. So a mixture of an attractive and aversive odorant could give a neutral response. Now, this wasn't true for all mixtures of attractive and aversive odorants, and furthermore, the effect could vary depending upon the attractive odorant used. So it didn't seem like a simple mutual cancellation of positive and negative signals, but we're not really sure what's going on. But interestingly, to our surprise, both a TAAR ligand, TMA, and a common odorant, PPA, were able to block aversion to the fox predator odor, TMT, suggesting that this blocking ability can even extend to a behavior of potential importance to the animal's survival. Luis further found that in animals lacking TAAR5, the TMA receptor, TMA was no longer able to block aversion to TMT. So this suggests that signals derived from one receptor, TAAR5, can act within the brain to block predator odor signals that cause aversion. So, excuse me, altogether, these studies suggested that innate odor-induced behaviors can be context-dependent. In like odor perception, they can be influenced by the combination of co-activated receptors in the periphery. They further suggest that signals derived from a single receptor in the nose can block innate odor aversion or be required for that blocking. And finally, that instinctive behavioral responses to odors can be profoundly influenced by interactions that occur in the brain among signals derived from different receptors. So we really want to know how this is occurring. And Jung Lee, who's here at this meeting and is a postdoc in the lab, is working hard right now to explore where in the mouse brain the blocking occurs and how it occurs. So we're hoping that she can figure that out. Okay, so I'd finally like to turn to a different question, and that is, how do odors induce instinctive fear responses in mice? Kunio Kondo, another postdoc at our lab, decided to investigate this question, and to do so, to focus on corticotropin-releasing hormone, or CRH neurons, in the paraventricular nucleus of the thalamus. So it's well known that predator odors can induce instinctive fear responses in mice that include not only fear behaviors, but also surges in blood levels of stress hormones. And these stress hormone surges are reminiscent of similar responses in humans to when they experience fear or stress. Excuse me. So what Kunio wanted to figure out was what olfactory inputs are targeted to CRH neurons in the hypothalamus to cause fear responses. Now, predator odors can be detected either in the nose or in the VNO. Volatile predator odors are detected in the nose, whereas those detected in the VNO so far all appear to be proteins, such as proteins from rat or cat. So with the detection of volatile predator odors in the nose, signals from the nose travel through the main olfactory bulb to the olfactory cortex. Now, the olfactory cortex is not a simple area, as Li-Chin Luo knows from his studies on the mouse olfactory cortex and looking at inputs to different parts from different parts of the olfactory bulb. In fact, the olfactory cortex contains multiple different anatomically distinct areas, and the functions of those different areas are largely unknown. Signals from the VNO instead are targeted to the vomeronasal amygdala, which has two parts, the MEA and PMCO. So what Kunio wanted to find out was whether there might be a specific part of the olfactory cortex that was involved in volatile predator odors detected in the mouse nose. So to do that, he and Zhonghua Liu first made some neurotropic viral tracers. Both were pseudorabies virus tracers, or PRVs, One is a new polysynaptic PRV, that is, that can travel across multiple synapses in a retrograde fashion. This virus, PRV-B177, has CRE-dependent expression that's very tight of an HA-tagged thymidine kinase, or TK. TK is required for PRV replication and spread from neuron to neuron. So this virus, when it infects a CRE-positive neuron, will replicate in that neuron. The HATK is irreversibly expressed after that, so it can travel retrogradely across a chain of connected neurons. The other virus that they made is a monosynaptic PRV, PRV-B316. This virus has CRE-dependent expression of GFP, but lacks TK entirely. They also made a lentivirus with CRE-dependent expression of HATK. So if both of these viruses infect a CRE-positive neuron, the HATK produced by the lentivirus can allow replication of the PRV and thereby allowing it to cross one synapse. But the lentivirus cannot cross the synapse, thus the PRV can't go beyond that first synapse. So Cuneo, to look at neurons upstream of CRH neurons in the mouse brain, Cuneo injected these viruses into the PVN of CRH CRE mice, which express CRE in CRH neurons. And these studies showed that using immunostaining to detect virus-infected neurons, these studies showed that there are multiple areas of the brain that contain neurons that transmit signals to the CRH neurons. It appears that 31 different brain areas can transmit signals directly to CRH neurons. That is, they're one synapse upstream of the CRH neurons, and the polysynaptic PRV results suggest that there are additional neurons in additional brain areas that are 2 or more synapses upstream of the CRH neurons. So the CRH neurons are gathering information, presumably about different aspects of the animal's physiology and brain function from multiple different brain areas. Now, the only area of the olfactory system that was labeled with the monosynaptic PRV was the MEA in the vomeronasal amygdala, suggesting that there are neurons in the MEA that can transmit signals directly to CRH neurons. But we did see neurons in 5 of the 11 different areas of the olfactory cortex that were infected with PRV on day 4 after injection of the polysynaptic PRV. Excuse me. So we think that these neurons are likely to be 2 synapses upstream of CRH neurons and thus send signals through a relay, we don't know where, in order to finally reach the CRH neurons. Now, the instinctive fear responses are similar among animals, suggesting that they might be controlled by hardwired neural circuits. One possibility is that those neural circuits involve spatially invariant subsets of neurons in the brain, such as clusters of neurons at particular sites. To explore this idea, Cuneo charted the exact locations of neurons upstream of CRH neurons in the different higher olfactory areas. And for the most part, the neurons were highly distributed. So there was no evidence, there was certainly no evidence at all of clusters of neurons sending information to CRH neurons. He did see some bias in a couple of areas. So there were more cells in the posterior piriform upstream of CRH neurons and more in the posterior part of the ampere amygdalo-piriform transition area, and in the one quadrant of the MEA in the vomeronasal amygdala. But for the most part, the cells were highly distributed. Now, another question was whether the neurons upstream of CRH neurons in these olfactory areas might be excitatory glutamatergic neurons or inhibitory GABAergic neurons. And to address this question, Cuneo infected the CRH neurons with the PRV, and then he did double labeling for markers of glutamatergic or GABAergic neurons, that is VGLUT1 or 2 or GAD1 or 2. And these experiments indicated that in the olfactory cortex and also in the PMCO, in the vomeronasal amygdala, most or all neurons upstream of the CRH neurons are glutamatergic neurons. And few or none are GABAergic. But in the MEA, in the vomeronasal amygdala, some neurons were glutamatergic and other ones were GABAergic. Since the MEA can send signals directly to CRH neurons, this suggests that there may be neurons in the MEA that can directly activate or directly suppress CRH neurons in the hypothalamus. However, remember that in the olfactory cortex, those neurons upstream of the CRH neurons are probably 2 synapses away. So although they're glutamatergic neurons, they could conceivably activate neurons directly upstream of CRH neurons that would in turn either suppress or activate CRH neurons. So we don't know for sure what effects these neurons upstream of the CRH neurons have based on these data. Excuse me. Okay, so remember that Cuneo saw neurons upstream of CRH neurons in 5 of 11 areas of the olfactory cortex. This suggested that CRH neurons might receive redundant input from different parts of the olfactory cortex about predator odors. To explore this question, what he did was to expose animals, virus-infected animals, to 2 different predator odors detected in the nose, TMT and bobcat urine. And then he looked at PRV-positive cells across the olfactory cortex to see whether there were PRV-infected cells that were activated by the predator odors. And for this, he used, as a neural activity marker, nuclear arc mRNA. And what he found was that there was a significant activation of PRV-positive neurons upstream of CRH neurons in only one area of the olfactory cortex, the ampere, amygdalo-piriform transition area, a very small and minor area of the olfactory cortex. And in fact, when he told us that, we said, the what? Because we hadn't really considered that before because it was a very minor area. So to investigate whether the ampere is actually involved in predator odor responses, Cuneo used a chemogenetic method with DREADS, which you'll hear more about later today, that is designer receptors exclusively activated by designer drugs. These are mutated G protein coupled receptors that can be activated only by pharmacologically inert ligands such as clozapine and oxide or CNO. There are 2 flavors of DREADS shown here. One is an activating DREAD, HM3DQ, which activates neurons through the GPCR pathway, and another one, H4MDI, which is inhibitory DREAD, which silences neurons by acting through a different intracellular pathway. So what Cuneo did first was to ask whether activation of the ampere by a DREAD or other areas could cause an increase in adrenocorticotropic hormone, or ACTH, which is one of the stress hormones that is controlled by CRH neurons. So what he did was to inject a adeno-associated virus encoding a CRE-dependent, with CRE-dependent expression of the activating DREAD into the ampere, the posterior piriform, or the MEA and the vomeronasal amygdala, and then measure blood levels of ACTH, the stress hormone. And what he found was that activation of the DREAD with CNO, either in the ampere or MEA, could cause a surge in blood levels of the stress hormone, ACTH. This was not true in the posterior piriform. So this suggested that neurons in both the ampere and the MEA can increase plasma ACTH, mimicking an instinctive fear response. But remember, the ampere is in the olfactory cortex that gets information about volatile predator odors, whereas the MEA is in the vomeronasal amygdala, which gets input from the VNO. And we wanted to know what happened in the case of volatile predator odors detected in the nose. So Cuneo next asked whether the ampere is required for predator odors detected in the nose to cause an increase in blood levels of stress hormone. And for this, he injected the ampere with a CRE-dependent adeno-associated virus that silences neurons when it's activated by its ligand CNO. And then what he did was to, he silenced the neurons in the ampere, then he exposed the animals to volatile predator odors, either TMT or bobcat urine, and then he asked what happened to the predator odor-induced increase in blood ACTH. And what he found was that when he silenced the ampere, there was a striking reduction in the ability of these predator odors to induce these blood level increases in ACTH, characteristic of an instinctive fear response. And consistent with this, he also saw dampening of the predator odor-induced activation of CRH neurons, and also the dampening of predator odor activation of the neurons in the ampere, which was, by the way, similar in extent to the dampening in the surge in ACTH in the blood. So these studies indicated that the ampere plays a key role in the stress hormone response to volatile predator odors. And this is remarkable if you consider that the ampere is a very minor area of the olfactory cortex, which actually occupies less than 5% of the total structure. As I mentioned earlier, the predator odors can induce an instinctive fear response that includes particular behaviors, as well as increases in stress hormones. And one of those behaviors is immobilization or freezing behavior. And Cuneo found that when he silenced the ampere, although it had a dramatic effect on stress hormone responses, it did absolutely nothing to the ability of predator odors to induce freezing behavior. So this suggests that the behavioral and hormonal response to predator odors, that is, the two arms of the instinctive fear response, the physiological and the behavioral, may involve different parts of the olfactory cortex. And consistent with this idea, Root and Axel actually provided evidence that another small part of the olfactory cortex is important in the freezing behavior of TMT in response to TMT. So what we'd like to do now is find out more about how these neural circuits that control fear function. We'd like to know that there are molecular signatures. You heard a little bit about that already from the fantastic work going on at the Allen Institute. We'd like to know whether there are neurons with particular neural signatures in the olfactory bulb or the olfactory cortex or other parts of the brain that are involved in the fear response. If so, these molecular signatures, the genes encoding them, can be used as tools to explore how those circuits actually function in the living animal. I'd finally like to acknowledge the really wonderful people that I've had the privilege to work with over the many years. These are postdocs and students who've been in my lab. Carrie Ressler, Susan Sullivan, Hiroaki Matsunami, Bettina Melnick, and several others did all the early work on inputs to OR inputs to the nose and bulb and found some of the receptor families. Steve Lieberlis also found the TAARs and the FPRs. Naresh Hanchate did the recent work on single-cell transcriptomes in the developing OE and OSNs. Lisa Horowitz did the study in macaque and with human TARs. And Luis Saraiva did the studies of OR TAR functions and odor blocking. Zhonghua Liu and Kunio Kondo prepared the new PRVs, and Kunio Kondo did all the studies of fear circuits in mice. So thanks very much, and I'd be happy to take any questions you may have.
So we have microphones on either side here if you want to put your hand up. Another heads up, we're also heading into a short coffee break. So those of you who are desperate to get to the coffee first, it's just outside here on your right. But considering the tour de force we just heard, you might want to sit and listen to some answers to some questions first. But up to you. Yeah.
That was a great talk. Thank you very much. My question was from the early part of your talk. It was really interesting to see the changes in gene expression going from early in development to late in development. And I was wondering if you could maybe just kind of speculate about what those observations mean. Do you think maybe as a necessary step from expressing only a single OLFR, it's required to start off expressing many? Or do you think perhaps instead there's maybe just a different strategy early in development? Maybe it's for the particular odors that need to be detected early versus late in life, that there might be a different kind of olfactory strategy early versus late?
Yeah, that's a very interesting question, whether those multiple OLFRs are actually functional at that point. They may not be. The transcript levels are extremely low at that stage, but we can't rule it out. And there were some very reports from many years ago that suggested that the immature neurons might, or in the neonate, that some of the neurons may be more broadly tuned and recognize more ORs, but more odorous, but we don't know if that's the case. Now, one analogy that comes to mind is in the case of immunoglobulin gene rearrangement. And there, when we saw these results, I remember in the 80s, because I was always interested in immunology, the finding that that multiple V gene segments were transcribed before the rearrangement occurred. So before, I mean, in that case, before the choice was made by a particular gene rearrangement event. And when I looked at some more recent work, it suggested that that's necessary for the event. So it could be to occur, the recombination to occur. And so if you block transcription off a particular, in this case, the J segment, you cannot actually get rearrangement. So it could be that this just reflects an open chromatin state. It's meaningless, and it just reflects an availability for that particular gene to be chosen. Or it could be that a particular gene manages to capture a particular set, an important set of transcription factors present at a very low level, and that allows its higher level expression. But we really don't know. But certainly this excludes the possibility that there's, which has been proposed, that all the genes are completely repressed and only one is de-repressed.
Question over there?
Giovanni from Synovion Pharmaceuticals. Wondering how you're kind of integrating some of these things, and are you thinking at all about post-traumatic stress disorder? How there may be some overlap, maybe not just the odor.
You know, there could be. I don't know very much about it, but apparently there is dysregulation of stress hormone levels in PTSD, as well as in major depression. So that's one avenue we'd like to explore, at least in thinking about it in the future, and see if there's something that we could do that could provide insight into these problems, because we do have all these upstream areas that are sending information to CRH neurons.
Or are you thinking more about aromatherapy to overcome that fear response?
I wasn't thinking that. But
Okay.
It could work.
Well,
Never know.
actually, Un, who I mentioned is at the meeting, is actually working on odor blocking of stress hormone responses. And so we'll see. We'd like to know how that happens.
Yep.
You showed nicely that the small area of olfactory cortex has this, remarkable behavioral role. I wonder whether the organization of different sort of innate odors of innate meanings, there is any spatial organization or anything which one can discern in olfactory bulb already, like the TAAR receptor targeting area. Do they specifically target to one particular area? How are they related to? And also the predator odors, their representation in the olfactory bulb.
Well, in the olfactory bulb, as well, first of all, the TAARs, neurons expressing TAARs do project just to a particular part of the dorsal olfactory bulb. And I should mention that Sakano and his colleagues have provided evidence in the past that the dorsal bulb may play an important role in innate responses, particularly aversive responses and fear responses. And Li-Chin Liu also has evidence that there's one part of the olfactory cortex that may be biased to receive input from the dorsal bulb. Although, again, it's very highly distributed across the olfactory cortex, as far as we know now anyway. So is there a particular part of the bulb? I think the Sakano lab also has evidence. From their work, there are many receptors for TMT. And there may be several involved in the fear response. But he hasn't published that work yet.
So one more question before we let you loose on the coffee.
Hi. So I know in the insect system, there's been some work on the structural basis of the tuning of receptors, but I just haven't really kept up on this. Is there any work on the structural basis for the tuning of odorant receptors in the mammalian system?
Well, there has been work doing site-directed mutagenesis and to look at the effect of that, looking at the tuning of individual receptors expressed in tissue culture cells and then making slight changes. And yes, there's certain, the extent of tuning, no, but the ability to recognize different odorants, yes, single changes can influence that, single amino acid changes.
Okay, so I'd like to have us thank Linda one more time and off to coffee. I would also like to remind the people at the back that snuck in during the talk that there's lots of seats down at the front.